After studying this module, you shall be able to know why biochemical reactions occur only by enzyme catalysis, free energy changes accompanying biochemical reactions, specificity of enzyme substrate reactions are the basis of their catalytic power. Let me introduce you to this module. All biological reactions essentially require enzymes. This is because substrates or reactants are stable and in order to change to products have to pass an energy barrier in the form of activation energy. Just as chemical reactions require catalysts, biological reactions require enzyme catalysts. Unlike inorganic catalysts, enzymes can enhance reaction rates by over 1,000,000-fold and that too at ordinary room temperatures and atmospheric pressure. This enormous increase is mainly due to the specificity of binding of enzymes to their substrates. The binding energy released due to specific interaction of the enzyme with its substrate provides in a major way the energy required to overcome the activation energy barrier which prevents substrates from undergoing reaction. Furthermore, due to the enormous diversity of substrates in biological systems, thousands of enzymes are therefore present which are able to selectively bind and enhance the reaction rates. A study of enzyme catalyzed reactions is incomplete without first considering the energetic or free energy changes accompanying biological reactions. Free energy changes accompanying enzyme catalyzed reactions. Enzymes accelerate key reactions which include energy transformation from one kind to another. For example, solar energy is converted into chemical bond energy in the form of sucrose by the various enzymes involved in photosynthesis by green plants. The bond energy in sugars like glucose are broken gradually by the enzymes of metabolism and trapped into high energy biological currency called ATP or adenosine triphosphate. Biological systems then use the chemical energy released by hydrolysis of ATP to drive another set of reactions such as muscle contraction, biosynthesis of various macromolecules such as DNA, proteins and polysaccharides, and even light as exemplified by the fireflies, as well as high voltage releases by the eel fish to kill or stun its prey. Hence, energy transactions are integral to life processes which then are carried out basically by enzymes. Let us look at spontaneous and non-spontaneous reactions. A spontaneous reaction is one which is accompanied with a release in free energy. In other words, the difference in the energy between products and substrates is negative. Consider a reaction of substrates A and B giving rise to products C and D. The delta G or change in free energy of this reaction is given by the equation delta G is equal to delta G 0 plus RT log C D divided by AB, wherein C, D, AB reflect the molar concentrations of the substrates and products, wherein delta G 0 is the standard free energy change, T is the absolute temperature, R is the gas constant. However, in biochemical reactions, a modified delta G 0 prime is used for free energy calculations, which is generally at pH 7. Delta G 0 prime is determined by measuring the concentration reactants and products when the reaction is at equilibrium. Thus, at equilibrium delta G is 0. Therefore, the equation will be 0 equals delta G 0 plus RT log C D divided by AB, wherein delta G 0 is the standard free energy change, T is the absolute temperature, R is the gas constant. However, in biochemical reactions, a modified delta G 0 prime is used for free energy calculations because these reactions are carried out at pH 7. Delta G 0 prime is determined by measuring the concentrations of reactants and products when the reaction is at equilibrium. Thus, at equilibrium delta G is 0. Therefore, substituting 0 in the above mentioned equation, 0 equals delta G 0 plus RT log natural C D divided by AB. Again rearranging, delta G 0 prime is equal to minus RT natural logarithm C D divided by AB. Substituting K equilibrium equal to C D divided by AB in the above equation, delta G 0 prime is equal to minus RT natural logarithm K equilibrium. R is equal to 8.315 into 10 to the power minus 3 kilojoules per mole per degree, and T is equal to 298 Kelvin, that is 25 degrees centigrade. Any reaction having a negative change in standard free energy therefore can be defined as a spontaneous one, and vice versa, a reaction with a positive change in free energy is non-spontaneous. In biochemical reactions, often non-spontaneous reactions can be coupled with spontaneous reactions provided that the coupled set of reactions are overall negative free energy yielding, so that a given task such as a biosynthetic process can be carried out. Therefore, even if enzymes cannot change the spontaneous or non-spontaneous nature of reactions, the latter type of reactions can be circumvented by coupling with a free energy yielding reaction such as ATP hydrolysis. It must also be emphasized that enzymes cannot change the K equilibrium of an reaction, but can only hasten its attainment. Enzymes alter only the rate of reactions. This is possible because enzymes are able to reduce activation energy barriers, as will be discussed shortly. What is activation energy? Consider the free energy changes accompanying the progress of a reaction, which has been graphically represented for you. The y axis is the energy level of the reactants or products, and the x axis is the reaction coordinate. Here the reactants such as A plus B have a certain energy level. They acquire an energy which is equal to energy of activation EA, then they reach a peak, after which they can drop down to form products which are C and D, and the change in energy between the reactants and products is therefore delta E. As indicated in the graph, the reactants A and B first have to attain an energy equal to EA or activation energy to become a transient transition state before the products C and D can be formed. Although as indicated in the graph, the energy of C and D is lower than that of A and B, and therefore the reaction is spontaneous, the energy barrier must be met. Enzymes are known to bind their substrates specifically, and evidence is gathering that they actually bind the transition state of their substrates, which is a high energy and transient state. The specificity of interaction of the transition state with the enzyme leads to the release of binding energy, which forms the basis of reduction in the energy of activation. How are enzymes able to release large amounts of binding energy? Details of these are explained in the next section. Let us consider the binding energy of enzyme catalyzed reactions. In an enzyme catalyzed reaction, the reaction velocity changes with the substrate concentration as indicated in the graph. The x axis has the substrate concentration and the y axis is the velocity. At lower concentrations of substrate, velocity is proportional to substrate concentration. Namely, you can see that the graph is linear, but at higher substrate concentrations, the velocity reaches a saturation or Vmax value. This type of rectangular hyperbolic curve suggests that enzymes bind to substrates in order to catalyze reactions and show saturation kinetics at higher substrate concentrations. As indicated previously, enzymes bind specifically to substrates and that releases binding energy which brings down the activation energy barrier. Enzymes fold into unique three-dimensional conformations which include clefts to bind the substrates surrounded by key amino acid residues which bind the substrates as well as participate in catalysis. The extraordinary specificity of binding between enzymes and their substrates can be appreciated in the stereospecificity and geometric specificity. Most biological substrates are either chiral or prochiral. Enzymes which bind to these sugars cannot bind to the L form of the same sugar. This exquisite stereospecificity is exemplified by the action of an enzyme alcohol dehydrogenase which catalyzes the reversible oxidation of alcohol to acetaldehyde, which is accompanied by the reduction of the enzyme's coenzyme NAD plus to NADH and H plus. NAD plus and NADH are the oxidized and reduced coenzymes respectively of the enzyme alcohol dehydrogenase. Nicotinamide adenine dinucleotide is represented as either NAD or NADH. These molecules have a prochiral center to which a hydride ion can be transferred or removed as indicated. Even ethyl alcohol has a prochiral center on carbon 2 from which a hydride is transferred to NAD plus during its oxidation to acetaldehyde. Elegant labeling experiments using the deuterium isotope of hydrogen indicated that for one class of alcohol dehydrogenases, if a dihydride residue was removed by oxidation, the same D residue was transferred back to acetaldehyde from NADD in the reverse reaction. This would only be possible if the enzyme bound alcohol and NAD plus in such a way which is stereospecific as indicated in the figure. Here alcohol has two hydride ions or two hydrogen atoms on its carbon 2. In the experiment, one hydrogen has been replaced by the heavier isotope deuterium. During the reaction of alcohol to acetaldehyde, which is oxidation, the deuterium has been transferred to the nicotinamide group of the coenzyme, namely to form NADH, but rather NADD. Now let us look at another kind of stereospecificity, namely binding to transition state. Enzymes use a variety of mechanisms for catalysis by virtue of having a large number of different amino acid residues around the substrate binding site. However, the most effective mechanism appears to be that enzymes bind to the transition state of the substrate transiently which is a high energy state. The transition state can break down to give our products or alternatively dissociate to give back the substrate. Breakdown or dissociation is decided by the free energy change. For a spontaneous reaction, a larger amount of free energy is released when the transition state breaks down to form the product. Enzymes, in binding specifically to their substrates, actually distort the latter, which causes strain in certain bonds so that they break more easily and new bonds are formed. Due to the distortion of bonds, the enzyme actually binds to the transition state of the substrate, which is already high in energy, and hence it can break down very easily to form products, which indicates the free energy changes accompanying the progress of a reaction, which has been plotted on the x-axis. Because the enzyme is binding to the transition state of the substrate, the energy requirements in terms of activation energy come down. So therefore, it appears that by binding to the high energy state or transition state of the substrate, the enzyme is now showing a different pathway for the reaction, wherein the activation energy has been lowered and therefore the substrates can easily pass through the energy barrier and then form the product. Observe also the schematic representation of the way the substrate is binding to the active site. The enzyme, in order to bind the substrate, undergoes a certain amount of change. The substrate also, in order to bind to the substrate binding site of the enzyme, undergoes a little bit of distortion. In other words, the enzyme also changes shape slightly as the substrate binds, and so does the substrate too. This really means that certain bonds of the substrate are either getting loosened or getting strengthened, and that is why the substrate is now acquired a high energy, or is sometimes also called as a transition state. The products which are formed by this interaction is observed as 2. So therefore, it means that the substrate, which was one molecule, has now become 2 products. The cartoon again tells you that the enzyme binds and stabilizes the distorted substrate, which is already a high energy transition state, and therefore reduces the activation energy barrier. Let me summarize for you what you have learnt in this module. Enzymes catalyze biochemical reactions by increasing the rate of reactions by more than 1,000,000-fold at ordinary room temperature and atmospheric pressure. Enzymes cannot change the thermodynamics of reactions, that is, free energy changes, spontaneous reactions or equilibrium constants. Enzymes can speeden up attainment of equilibrium by decreasing activation energy barriers. The activation energy barrier is reduced in enzyme catalyzed reactions by the enzyme binding specifically to transition states of the reacting substrates and releasing binding energy.